Here we have a specific example of hormonal control within the male reproductive system. So there's the hypothalamus. The hypothalamus, once puberty sets, is going to be releasing this gonadotropin releasing hormone. That gonadotropin releasing hormone will act upon the anterior pituitary, and the anterior pituitary will release follicle stimulating hormone and luteinizing hormone. These two particular hormones will act on Sertoli cells and Leydig cells, respectively. And as a result of these hormones, we ultimately are capable of achieving spermatogenesis. Now, when it comes to the Sertoli cells, these are going to be producing inhibin as well, which we've discussed with respect to the HPG axis, can serve within a negative feedback loop in order to inhibit that process or place some form of regulation on it. There's also the production of testosterone. And this is kind of a downstream thing. There is something in between here that's not in the image, and we won't discuss it for principles, but it is something that those that have taken or will take anatomy and physiology 2 will discuss. But ultimately, testosterone will help with the process of spermatogenesis, but can also exert a negative feedback loop, acting upon the anterior pituitary, or based on this illustration, acting upon the hypothalamus to turn this response off. As a reminder, for those that have had me in earlier versions of this course, you'll know that inhibitory signals should be a horizontal line, should not be a red arrow, in order to indicate that there is an inhibitory role that's being played here. And normally an arrowhead should be reserved for stimulatory roles. But ultimately, this is showing us the HPG axis, specifically within the male reproductive system, and how the stimulation of these different components within the endocrine system help to achieve spermatogenesis, which takes place within the testes and was discussed with respect to the anatomy in an earlier voice thread. When it comes to the female reproductive cycle, you'll find that there's a little bit more with respect to information and detail because, as mentioned earlier, the process is a little bit more complex. And when it comes to the cycles, they are going to be defined by what type of changes we're seeing. So the ovarian cycle, ovary changes, uterine cycle, changes within the uterus. And when you're looking at these cycles, think about how they are working with one another, how the changes are specific to the ovaries as well as the uterus, but how these are ultimately helping to support the idea of preparation for a tentative pregnancy, but then also how the body responds if that event does not take place during a particular cycle. The HPG axis is seen here as well. So the level of organization and expectation is consistent with that of the male reproductive system, with the onset being during puberty. So there will be the hypothalamus, which will result in the release of gonadotropin releasing hormone, which will stimulate the anterior pituitary to release FSH and LH. And there will be negative feedback loops that are seen that can help to inhibit gonadotropin release as well. And so the concentrations of estradiol can exert negative feedback on the anterior pituitary as well as the hypothalamus, and that would lead to inhibition of the downstream release of FSH and LH. Now, when it comes to this process, there are going to be fluctuations and surges that are seen within the luteinizing hormone, and this will have an impact on different types of cycles that occur within the female reproductive system. And so we'll go through those together. When it comes to panel B, this is looking at shifts in the pituitary gonadotropins within the blood. So it's looking at luteinizing hormone and follicle stimulating hormone. And so, as the name implies, the follicle stimulating hormone as well as the luteinizing hormone will help to stimulate the follicle to grow. Now, when it comes to the development of oocytes, they're found at all different stages of development. And through these fluctuations in gonadotropin levels, only the dominant follicle will essentially be pushed towards the process of ovulation. And the specific details of that we're not going to go over in this particular course, but once that dominant follicle is chosen or determined, it in and of itself can produce high levels of hormone, and that can help to kind of ensure that none of the other oocytes that are present within the ovary are released too. And so there is a surge that is seen within the luteinizing hormone, and that is going to promote ovulation. And that surge will promote the transformation of the ruptured follicle into the corpus luteum that we discussed earlier. So we can see that around day 14, we have this peak, and that triggers ovulation. And for those that are planning pregnancies, having knowledge about when that individual's day 14 will be can help to increase the likelihood of getting pregnant successfully, or if those are avoiding it, knowing when that ovulation day can have an opposing effect if avoiding pregnancy is the goal. When it comes to the illustration, this is also showing a 28-day cycle, but it's entirely possible for an individual to have a cycle that is less than 28 days or more than 28 days. So this is just an example that's often utilized in biology textbooks, but it doesn't apply to everyone because everyone is different. At this point, we've already gone over how the follicle changes and progresses. What you want to do with these particular images is now start to overlay how the gonadotropins will fluctuate or change and how that correlates with modifications of the oocyte and the surrounding follicles. So in looking at these, you want to think, okay, well, when I have that surge, what takes place, which I mentioned in the slide before it, when you are looking at this particular image, we're thinking about how the follicle is changing again, but now we're seeing its relationship with that of ovarian hormones. So taking a look at estradiol and progesterone, seeing how at the beginning of this process, their concentrations are pretty low, but then we start to see estradiol go up. We see a little bit later on, so kind of lagging behind, progesterone going up, and the role that each of these are playing in the different events as they relate to the ovarian cycle and the uterine cycle. Here we have the uterine cycle. So we first talked about how the follicle develops. Now we'll talk about how the uterus kind of cycles and changes. And again, we're still thinking about the role that hormones are playing. So hormones coordinate the uterine cycle, hormones coordinate the ovarian cycle. So when it comes to the uterine cycle, we'll go through these specific phases. So you have the menstrual phase. You can see kind of just the change in the tissue that's seen here. So the uterus is going to shed the majority of its endometrium. And at this point, the hormones are relatively low. And that's important to keep in mind because the endometrium depends on hormones. So it's hormone dependent. And so this decline in hormone levels here will serve as an indicator that the tissue here, the cells here will slough off. So they'll detach from the uterine wall, and that will result in bleeding. And this tissue will exit through the vagina as menstrual flow. After the menstrual phase, you have the proliferative phase, and proliferation, we're thinking about growth. So we're going to see a rise or a thickening of the endometrium. So we're rebuilding here. And this will be influenced by changes in estrogen. So estrogen levels will rise during the proliferative phase. And this helps towards the production of a new functional layer. So you have your functional layer here, you have your base layer here. So we're contributing towards the proliferation of a new functional layer during that proliferative phase. So when it comes to this process, the glands that are found here are also going to enlarge, and we're going to see an increase with respect to vascularization. And there will also be an induction for cells of the endometrium to synthesize progesterone receptors so that they can respond to progesterone levels. There's also a kind of change with respect to the cervical mucus that takes place here, and that's really to help with the movement of sperm through the reproductive tract of the female. Now, when it comes to this process, kind of our point here in terms of release is going to be ovulation. So ovulation is going to mark the end of the proliferative phase, and that's going to be in response to the LH surge that was mentioned in the earlier slide. So in response to that luteinizing hormone surge, we're going to induce that weakening or that transition to rupturing, and that will lead to ovulation at that 14-day mark within the cycle. After we have release of that oocyte, we enter into the secretory phase. So the endometrium has thickened here, so we can see this continued progression upwards. It's accumulated a ton of glycogen, and this is all in preparation for the process of possible implantation. And so there will be secretion of nutrients into the uterine cavity with the goal of helping to sustain an embryo if an embryo is indeed implanted. Now, progesterone levels are going to rise, and cervical mucus consistency is going to change. So usually what happens is if we have that successful implantation, then cervical mucus consistency becomes more viscous, and something called a mucus plug will form. And that's meant to block any additional entry of sperm. And it's also beneficial because it should be in kind of blockade to pathogens or foreign material. So it's meant to serve a protective purpose. So if fertilization has not occurred, then what we would expect here is a degeneration of that corpus luteum. If we don't have that endocrine gland, then progesterone levels are going to decline. And then that is going to be consistent with less hormonal support for the endometrium, which comes right back around to that idea of sloughing off of the weakened tissue. And so the uterine cycle will begin again. So if pregnancy does not occur, then we head back into the menstrual phase because we no longer have any of that hormonal support once that endocrine gland has been degenerated. Keep in mind that in the explanation of what's going on within the uterine cycle, I'm saying estrogen, but estradiol is the predominant variant of that hormone that presents in humans. So I'm using it interchangeably here. And when it comes to the different layers that are seen within the uterus, there is the functional layer, which makes up the majority of it. And then there's like a basal layer, which I referenced earlier. You don't really get a proper sense of that using this particular illustration, and it's not something that you would be necessarily responsible for, being able to tell the difference between the functional layer and the basal layer on any type of exam. It's really just more of a comment that's relevant to the anatomy of the uterus during this process. Now that we've gone over the menstrual cycle, I would just like to think about the relationship of the ovarian hormones while these events are taking place. So what we find is initially both estradiol and progesterone levels are relatively low, but then they start to shift over time. And when we relate this to the rise, we start to see the rise in estradiol taking place during the proliferative phase. So what's taking place during the proliferative phase? So we have the endometrial tissue that was lost in that last menstruation being rebuilt. And so estrogen is helping to stimulate mitotic division of the base layer, so the lowest level, in addition to regrowth of blood vessels. And that's going to help to regenerate the functional layer, so the layer that's higher above that process. So estrogen is going to achieve or help with the process of the proliferative phase, and it also stimulates endometrial cells to produce progesterone receptors, and that's what prepares them for the secretory phase that follows. And when it comes to that rise or change with respect to progesterone, we can see that there's an overlap with the secretory phase, and that can also be connected to the corpus luteum. After a certain amount of cycles, human females will go through the process of menopause. And menopause is essentially cessation of ovulation and menstruation for at least 1 year. And the thought is that it might allow for the mother to provide better care for her children and grandchildren. I'm not one for speculating without any type of, concrete data per se as to why this would be the event. So I'd rather focus on it from a biological standpoint. And when it comes to this process, what's essentially happening is that we can't achieve the cyclical hormone changes anymore. And so the feedback loop that occurs initially during the time in which women are going through their reproductive abilities, it's no longer there. So 20 is considered to be the peak with respect to reproductive abilities, and at 30, the quality begins to decline. And by the time an individual is at about age 50, there's a really small amount of eggs that are left. Recall that they're in the process of oogenesis, or production of the primary oocyte. All of them are produced prior to birth, and there's no additional ones that are made, right? So there's a finite amount that are available. Now, in thinking about menopause, there are different changes that occur as a result of fluctuations in hormone levels. So there's going to be low levels of estrogen. This can change vaginal secretions. It can also result in vasodilation of skin blood vessels, which is what results in hot flashes. There can be loss of bone mass due to estrogen levels decreasing. So it's not, as simple as ovulation and menstruation cease, but there are a lot of things that result due to menopause, and that really has to do with low levels of estrogen. Now, for our purposes, really what you want to think about is if there is cessation of ovulation and menstruation, what is that telling us about the signals that would have been there to help promote that process? When it comes to the human sexual response, I'll just focus on the 4 phases, which are the same in both male and female, but there are differences that exist within these phases. But for this particular class, I'm not going to go through those distinctions. So when it comes to two major reactions that occur within both sexes, there is vasocongestion. So this is the swelling of tissue with blood. And then there's also myotonia, which is increased muscle tension. So let's just briefly go through these 4 phases just for the sake of how we define them. So it is the excitement phase that's characterized by that vasocongestion and the myotonia. But in addition to those things, there's also an increase in heart rate, an increase in blood pressure, breathing, right? So pulmonary ventilation would be the more, scientifically accurate way of describing that. And so there is a broad spectrum of ways in which the excitement phase can be initiated. And, because that varies from person to person, I don't think that there's, a real benefit to listing the mechanisms of excitement, right? But essentially, the physiological response is what I've mentioned earlier. During the plateau phase, things that are variable, like the pulmonary ventilation or heart rate, will remain at a pretty high level, if not increase slightly, prior to entering the orgasm phase, which is a relatively intense but short-lived phase that is then followed by resolution. And by the time we are at resolution, cardiovascular and respiratory functions are returning to normal. Now, the specific behavior or responses that are going to take place will vary between the male and female sexual response. So I won't add in the details because it will add more content to know, but just knowing what these 4 phases are, knowing that these 4 phases apply to both male and female reproductive systems, but there are some slight differences that are seen there that we just won't get into, that's sufficient as it relates to this material. Concept 46.5. In placental mammals, an embryo develops fully within the mother's uterus. So this is internal fertilization, where all of the development stays internal, and then there is utilization of a placenta. So there's a blood supply that helps to provide that nourishment. This is a concept that we talked about in the earlier voice thread slides. So there are specific events in which the egg transitions into that of an embryo, and we'll go through some of those major events within this concept or within this module. So we'll start off in the pre-embryonic period. And in this image, we're going to walk through the stages of fertilization. So at this point in the journey, sperm has been released and it's in search of an egg to fertilize. And there's tons of sperm that's released during ejaculation, but very few of those are successful in making it to the uterine tube where a meetup can take place. Another thing to factor into this level of success or ability to achieve that meeting event is that that secondary oocyte that is released during ovulation occurs at just one uterine tube as we go through these cycles, which means that some sperm may be heading down the wrong uterine tube for that particular cycle and will never have an opportunity to interact with an oocyte that was ready for them, right? That the one that was chosen and ejected during maturation. So sperm is heading through the reproductive tract, and as it heads through the reproductive tract, it has to go through the process of capacitation, and this is what makes the sperm capable of penetrating the egg. So there's some changes that are occurring, and when it comes to sperm cells, they have cholesterol on the membrane, and that protects the sperm from releasing the enzymes that are housed within them too early. So we wouldn't want them to be released while they are within the male reproductive tract. That may cause some enzymatic breakdown of components there that should not be. But now that it's entering the female reproductive tract, it needs to be broken down. So the fluids within the female reproductive tract can help to assist with this process. Now, when we're looking at step 2, we have the acrosomal reaction releases enzymes from the head of the sperm. And when it comes to sperm movement through this female reproductive tract, it can remain viable within the tract for up to 6 days. So if a copulation event happens too early relative to release of that secondary oocyte, a fertilization event can't happen. At the same time, if ejaculation into the female reproductive tract happens a little more than half a day, it's like 14 hours post-ovulation, the egg's no longer going to be viable by the time the sperm has gone through this process of capacitation. So timing is really important for those trying to conceive, and it should be based around the days closest to ovulation. If avoiding pregnancy is the goal, that window in terms of avoiding copulation needs to be much larger because it needs to account for variations in events based on egg longevity, sperm longevity, variations within the ovulation window, and things like that. So it's really important to know the date of ovulation or the cycle or process in both scenarios, trying to conceive and trying to avoid conception. So we have the sperm cell. It has the ability now to utilize the acrosomal enzymes that are housed within there post-capacitation, and it's going to help clear a path through the granulosa cells in the zona pellucida that surrounds the oocyte. So the first sperm to reach the egg is not the one that fertilizes it. It just helps to clear a path for another sperm cell to hopefully successfully interact. Now, when it comes to the acrosomal reaction, we have exocytosis of the acrosome that releases those penetrating enzymes. And there are different enzymes in there. One example of an enzyme that's present is that it can help to digest hyaluronic acid that binds the granulosa cells together. Now, in supporting our initial discussions of meiosis, we know that we go through 2 rounds of successive division in order to generate haploid gametes. Fertilization will allow for 2 haploid sets to come together in order to produce a diploid set. So we would want to avoid anything that would increase the ploidy, such as 2 sperm fusing with an egg. That would create something that's triploid. And so we have mechanisms to thwart this process. So our 2 mechanisms to prevent what is essentially polyspermy, too much sperm resulting in a ploidy that would be inappropriate as far as genetic content is concerned, is a fast block and a slow block. So in the fast block, sperm binding is going to result in an influx of sodium ions, and sodium ion movement into a cell can result in depolarization of the membrane. And this depolarization is going to inhibit additional sperm attachment to the egg. Okay. Then there is the slow block. So sperm penetration can release calcium ions, which stimulates a cortical reaction. And that reaction causes a fertilization membrane to form, which creates a barrier between the egg and the zona pellucida. So in this particular example here would have been the sperm cell that successfully made its way into the secondary oocyte. And notice that as the sperm enters, only the head makes its way in, which was the genetic component. So it's leaving the midpiece and the tail on the outside of the cell. So this essentially means that any mitochondrial DNA that's passed along is inherited through maternal information rather than paternal. But some studies suggest that this may not be the case. But the way that it's currently described is because the mitochondria is found within the midpiece and the mitochondria does not make it in during fertilization, no paternal mitochondrial DNA is passed on. Now, when we're looking at the sperm nucleus, notice that it goes through a change from sperm nucleus to male pronucleus. So there is a swelling that occurs. There will be microtubules that sprout, and that's going to help to push the sperm pronucleus towards the center of the egg. Now, as a reminder, our secondary oocyte was paused within the process of cell division. So once we have a fertilization event, we can continue with the process of meiosis too. And we need to shed some of that excess genetic information, which is why we see this polar body here. So that's achieved by generating that much smaller polar body. And the oocyte will also swell, and that will result in a female pronucleus. There is then a mitotic spindle, so we see that indicated here, and that's going to form between the pronuclei and then eventually rupture, and that's going to allow for the chromosomes of the two gametes to get together as a single diploid set. The image provided here shows us of the early events that take place within embryonic development. So what we're looking at here is the process of cleavage, which ultimately leads to the formation of a blastocyst, and a blastocyst will eventually plant itself into the uterus. The process of cleavage is mitotic division, and it's mitotic division that happens in the first few days as the conceptus works its way down the uterine tube. When it comes to mitotic division as it relates to cleavage, this reads a little bit from our discussions in Bio 100 as it relates to mitosis. As a reminder, when you think about mitotic division, you have your parental cell, and then that parental cell is going to prepare itself in different ways in order to ultimately give rise to identical daughter cells, identical with respect to genetic content, identical with respect to the size of the parent. In this context, in the process of cleavage, the mitotic division is rapid, but there is no growth that takes place alongside division. So this first portion of embryo development, the goal here is to produce small cells. And so just as we take a look at its progression, we can see that the size of the fertilized egg that we started off with isn't changing, but the size of the cells that are found in there are getting progressively smaller. The benefit here is also another concept that you already have learned, and that is the process of surface to volume ratio, which is like a way beginning of Bio 100. And so producing these small cells helps to support a high surface to volume ratio. And having that high surface to volume ratio helps to enhance the ability of exchange. So there's exchange of nutrients, exchange of oxygen, exchange of waste that can take place more effectively by capitalizing on that relationship between surface area and volume. Now looking at these cleavage events, one at a time, we see that first cleavage division produces two cells. These cells are referred to as blastomeres, and the blastomeres will continue to divide, so there will be the 4-cell stage, the 8-cell stage, and we will reach by day 3 this morula stage, and this is really named off of a mulberry or a little mulberry stage because they think that it looks like a little berry-shaped cluster. And at this point, there's about 16 cells that are found within. Now sometimes the morula will split into two masses. As that fertilized egg heads towards implantation, so you can see by the time we're at the morula stage, we're very close to embedding into the uterine wall. Now, if that happens, if the morula splits into two masses and both of them are going to implant, what that results in are identical twins. Now, in contrast, for fraternal twins, that would result from the release and fertilization of two eggs. So identical twins, single egg, that will split into two masses through the process of cleavage, while fraternal twins is two eggs to begin with. So the release and fertilization of two eggs is what would yield fraternal twins, and those would be genetically distinct from one another. They're derived from different cells, and obviously two different sperm cells because it's the release of an egg and fertilization of that egg. Now, when you look at the morula, you also see that the perimeter is changing qualitatively, and that perimeter is the zone of oocyte. And at this point, there's regeneration breaking down. Once we have that breakdown, we have a blastocyst. Okay, so don't confuse blastomere with blastocyst. So we now are at the blastocyst stage. And the blastocyst is essentially a hollow sphere, and that is zoomed in on here for day 4. So you have this hollow sphere. This hollow sphere is composed of two cell types, and there are the flattened cells, and those flattened cells are the trophoblasts. And then there are slightly rounded cells, which are the inner cell mass. And the inner cell mass is relevant to the discussion of stem cells and the ability or inability of certain stem types to give rise to extra embryonic tissue. So it's just a callback and a connection to our stem cell discussion. So here we go through the process of implantation in which the blastocyst attaches to the endometrium, and the blastocyst is going to have trophoblast cells separating out into two layers. So you have the syncytiotrophoblasts and the cytotrophoblasts, and the syncytiotrophoblasts will form this fused multi-nucleate mass, so you can't really discern one cell from the other there. The cytotrophoblast, in contrast, will retain its cell-like properties using one cell from another. So that's the one that's directly above this lighter tan color. And when it comes to the cytotrophoblast, it's going to grow into the uterus, so kind of extending like roots. And in that process, it's going to start to digest the endometrial cells. And the endometrium is going to respond to this digestion by essentially growing over the blastocyst, covering it, but it eventually becomes buried into the endometrial tissue. During this time, the ear is going to grow the endometrium. It's going to produce these kind of blood-filled lacunae areas in which there will be intervillous space in the functional layer. So when the blastocyst is fully implanted and it's completely surrounded by uterine tissue. Here we're thinking